Hello, everyone, and welcome to another MOGCast. This one's entitled MOGAD Clinical Trials 101. So today we're speaking with Associate Professor Sudarshini Ramanathan from University of Sydney in Sydney, Australia, and Dr. Michael Levy, who's the Director of the Neuroimmunology Clinic and Research Laboratory at Massachusetts General Hospital. My name is Julia Leffler. I'm the Executive Director and co-founder of the MOG Project, and I happen to be a MOGAD patient myself. We'd like to thank the doctors and the MOG squad for their contributions to this MOGcast. I'm co-moderating the webinar today with Jenny Kazan-Rodriguez. Jenny?
Thank you, Julia. My name is Jenny Kazan-Rodriguez, and I'm the Australian Ambassador and Medical Professional Outreach Specialist for the MOG Project, UCB's International MOGAD representative, a primary school teacher from Western Sydney, and a relapsing MOGAD patient.
So this MOGcast is being recorded and it's going to be available on our YouTube channel. Please feel free to submit questions in our webinar Q&A area. Any unaddressed questions will be answered in a follow-up MOG blog. Jenny?
I would like to introduce our first sponsor of this MOGcast, Roche. Roche has grown throughout a 125-year history into one of the world's largest biopharma companies, as well as a leading provider of diagnostic tests across various common and rare disease areas. The company owns a long history of neuroscience successes with medicines and diagnostics helping patients across the disease spectrum in areas such as neuroimmunology, neurodegeneration, neuromuscular disease, neurodevelopment, and psychiatry. Roche's ambition is to preserve what makes people who they are and create a tomorrow where neurological disorders no longer limit human potential. To achieve this, the company collaborates with academia, scientists, patients, patient advocacy groups like the MOG Project, and people like yourselves, industry partners, and clinical trial participants. With their support, there is a collective responsibility for meaningful therapeutic advances for some of the world's most difficult to treat diseases. Roche's commitment in neuroimmunology is to develop safe and convenient medicines that reliably treat disabling diseases of the nervous system. Roche understands the impact of MOGAD on people's activities and their quality of life. Roche's goal is to further support the rare neuroimmunology and MOGAD community and collaborate on development of effective treatments and diagnostic tools to help tackle this debilitating disease and many others.
Thanks, Jenny. UCB, our other sponsor for this MOGcast, is driven to ensure that everyone can live the best life they can, as free as possible from the challenges and uncertainty of disease. This ambition fuels their purpose. It creates value in the lives of the people they serve now and into the future. Their research and development activity across neuroimmunology, immunology, and other areas where their expertise aligns with unmet needs is driven by the connection to the people they serve. Their unique and diverse perspectives serve as guides and inspiration as UCB explores the furthest reaches of science and medical knowledge. They listen to and learn from patients, ensuring that their work has the greatest possible impact by delivering differentiated solutions and create value that cannot be expressed in numbers alone. They seek out tools, technologies, employees, and partners that let them respond to patient needs with purposeful innovations that make truly meaningful impact. From the headquarters in Belgium to nearly 40 countries around the world, they pursue that ambition each day, promoting a culture of collaboration and curiosity that values diverse perspectives and backgrounds. They are committed to caring for each other, for their communities, for the planet, and for the people who inspire their work. Jenny?
We're delighted to introduce our first panelist, Associate Professor Sudarshini Ramanathan, who is a neurologist and clinician scientist with subspecialty expertise in neuroimmunology. She's a staff specialist neurologist in Sydney, where she looks after patients with autoimmune neurological disorders. She leads the Translational Neuroimmunology Group at the University of Sydney. Her research program is focused on understanding disease pathogenesis and improving the diagnosis and treatment of autoimmune neurological disorders, including MOGAD. In 2013, Associate Professor Ramanathan established and has since been the lead investigator of the Australasian MOGAD study group, which encompasses over 150 clinicians from over 45 centres in Australasia. This team currently evaluates a cohort of over 800 MOGAD patients.
We are also thrilled to introduce our other panelist, Dr. Michael Levy, who's an associate professor in neurology who was recruited to lead the research unit in the new division of neuroimmunology at Massachusetts General Hospital. His mission is to build a combined clinical and research neuroimmunology program to develop therapies for patients with autoimmune diseases of the central nervous system. Dr. Levy moved from Baltimore, Maryland, where he was on the faculty at Johns Hopkins University since 2009 and director of the Neuromyelitis Optica Clinic. Clinically, Dr. Levy specializes in taking care of children and adults with rare neuroimmunological diseases, including neuromyelitis optica, transverse myelitis, MOG antibody disease, and acute disseminated encephalomyelitis. In addition to 4 monthly clinics, Dr. Levy is the principal investigator on several clinical studies and drug trials for these conditions.
We are here today to discuss all things related to MOGAD clinical trials with our wonderful experts, where we will be tackling all the wonderings you may have regarding clinical trials in MOG antibody disease.
I just wanted to welcome everybody. We're really excited to get, going and highlight these important topics internationally. So I want to get started with a short presentation from Dr. Levy before we move on to our questions. Dr. Levy?
Yes, thank you, Julie. I just have 10 short slides that outline what basically a MOG trial is for new drugs that'll help prevent relapses in MOG antibody disease. And these were put together, thank you to Julie and other people for the wonderful slides. Here's the agenda, probably 2 slides on each topic here. What is a trial? Who's eligible to participate? What is the structure of a trial? The different phases of clinical trials? And what control do you have as part of the trial? Okay, so just to emphasize, first of all, on the bottom pink there, every single drug that's approved for use in the United States and probably in the world has been approved through a clinical trial design. There has to be some sort of scientific evidence that that medication works better than something else, that something else might be nothing. But you have to prove scientifically that it's better. And you can't just say, I feel better on it, and that's why it should be approved. You have to actually design a study that you can prove scientifically that it can't just be, that this group of people who know that they're on a drug say they feel better compared to people who are not on the drug. So you'll see how we design these trials in order to be scientifically more certain about these types of things. We also do trials for safety. We need to confirm that these drugs are safe in the long term, safe by blood test. So that's the other big reason to do a trial. It also depends a lot on what question you ask. So for a lot of people, I tell them about all the new drugs that are approved for NMO. And after I'm blue in the face with all the talking, they'll say, well, can you get me out of this wheelchair with these drugs? And I'll say, no, because all of those NMO drugs are designed to prevent the next attack, but they don't do anything for your old damage. And so they have a disappointed look on their face. And they say, but aren't these drugs approved for NMO? And I'll say, yes, but they're approved for a specific task within NMO. And same is true for all the MOG studies. We're designing these studies to do a specific thing within MOG. So they won't fix everything, but they'll hopefully fix the 1 or 2 things that we're designing the studies for. Okay, now this is the important part here is that these studies are designed so that we get really good scientific answers. If I tell the patient that they're getting a drug, there might be a placebo effect in their mind. So we want to try to be as blinded as possible. We call it masked, where I don't know if a patient's on a drug. Patient doesn't know if they're on the drug. They might feel different, but it could be placebo effect. We really want to try to keep things as scientifically clean as possible. And at the end of the study, everything is unmasked. And then we can look at the data and say, okay, this drug does or does not work. And it's really important. I mean, we have experience, and I could tell other doctors around the world how I treat MOG or NMO, but that might not be scientifically accurate. And we've had to change our practice many, many times after scientific studies have proven that we might be doing something wrong. So that's why it's so important to do these studies. We're not just doing it to make drug companies money. We're doing it so that we can be sure about what we're prescribing. Who is eligible to participate? Anyone who has the disease might be eligible to participate. There might be other criteria. For example, in the MOG trials, we're only taking relapsing cases, meaning you have to have your first attack, diagnosed with MOG, and then have had a second attack. Because that indicates to us that that person has an active disease process and we can intervene there. A lot of people with MOG have just one attack, never have another attack. And we don't really want to take those patients because maybe if we give them a drug and they never have another attack, it might just be the natural history of their disease. So we impose an additional criteria. So you have to have MOG, you have to have relapsing disease, and there might be more criteria. For example, if you have cancer, that's going to drive your immune system. We don't really want that to interfere with our scientific study because most people with MOG don't have cancer. So there might be additional criteria. Age might be a criteria. The FDA is really hesitant to approve trials in kids if not enough adults have been exposed to provide a safety signal. So oftentimes we have to do a study in adults all the way through first, and then do another study in kids. So all these reasons, there might be additional criteria that are imposed on top of just, do you have MOG or not? But of course, we never discriminate based on race, ancestry, sexual orientation, gender identity, or disability status. Although disability status sometimes in the NMO study, for example, in a couple of the trials, if you were already bedbound, you were not eligible for the study. So that sometimes is not the case. Okay, so how is a clinical trial generally structured? It's structured extremely well. Okay, there are hundreds of people involved. They make all these rules. Everything has to be perfect. Every I needs to be dotted. Every T needs to be crossed. Because at the end of the day, when you get all your data, you need to be 100% certain that that data is good. You might have seen in the news, there's a lot of scientific misconduct going around the news at even prominent universities. We can't have that because patients' lives are at risk. So we have to do the studies very, very, very carefully. And we need to make sure that all data is perfect. And if I forget to sign something, there's 3 people bringing my door down saying, yeah, I need to look at this and sign it. So that's how these are structured. So when you say, oh, why can't you be a little bit more flexible about this? There's no flexibility in trials. It has to be done perfectly. There's a whole team involved at every site. The principal investigator, that's me. I'm ultimately responsible for everything that happens here. But I have a whole team, coordinators and nurses, and pharmacists, and everybody else, radiology techs, everybody works together and they all have their responsibilities. And there's a protocol, and the protocol is basically the instructions. It's a manual. It's like this thick, has every single thing you can think of. What temperature to store blood samples at, how long they can be stored, how long the MRIs, all those details, they're all in the protocol. It's very, very, very strict. And at the end of the day, when you get your data and everybody follows the same protocol, you can be sure that the data is good. The enrollment criteria are also very specific. So I've had patients who were excluded from a study because they didn't test positive for MOG anymore. And they say, well, but you know I have the disease, doc. And I said, I know, but everything has to be so scientifically sound that there can't be any question at the end of the day. Then when the trial is done and the drug is approved, then we can be a little bit more flexible and apply some clinical judgment. But in the trial, it's strict. And a lot of that is to remove this thing called bias. A lot of clinical trials in the olden days when the doctor was like, yes, I believe, and they took the opinion of the doctors more seriously, were all flawed. Okay, so you can't trust doctors in this. It has to be scientifically sound, and that removes the bias. So the patients can't say, can't ask for any flexibility, and doctors cannot impose their opinion. It has to be scientific. And in some studies, you compare two drugs, and in some studies, you compare the drug to no drug. Usually in trials where nothing is approved yet, like in MOG, the comparator arm has to be placebo or no drug. And that worries people. It worries me. If I enroll a patient into one of these studies and I say, well, there's a 50-50 chance you get the drug, then they'll say, well, what happens to the other 50%? Well, they get nothing. They get saline or sugar pill or something like that. No real drug in the comparator arm, because we have to be certain about the effect of the drug. If we allow people in the comparator arm to use steroids or something else, that's going to confound the data. So I understand the ethical concerns. In the NMO study, they allowed some background therapy like mycophenolate to protect those placebo patients, and we got through the study. And at the end of the day, we finished the trials, and we have 3 approved drugs. Now there's this thing called randomization, which is where I don't get to decide which arm you go to. Wish I could. Wish I could put all my patients in the treatment arm, and then I don't know where their comparator arm would come from, but I can't do that. There's a randomization process. So if you enroll at our site, I can't promise you either way. I get it, the pharmacy will just send up a drug. I have no idea if it's saline or if it's a real drug, and you're randomized somewhere else, and it's completely random in terms of which arm you end up in. You might end up in the treatment arm, you might end up in the placebo arm. And you get the drug. And if it works, well, great. You could be responding to placebo. We don't know until the end of the study. And if you relapse, we still don't know what you had. But in all of the MOG and NMO studies, if you have one attack, you're done. Then you move out into an open label phase where we know for sure you're getting the drug. And you're out of the experimental phase of the study. We're not monitoring you as closely for a relapse as we were in the experimental arm. We're really going to send that data to the FDA for approval or not. Once you're done with one relapse, you're in that open label phase. You're getting drug for sure. We monitor you for safety and things like that, but we're not monitoring any placebo patients anymore. So that allows patients to get the drug for sure, but they have to go through that experimental phase where you might have one relapse. There's this thing called blinding. That's the masking process I told you about, where you don't know what you're getting and I don't know what you're getting. If it's a single blind study, I know what you're getting, maybe because I know the color of the drug or something like that, and I'm going to try to keep that to myself and not let it influence me. But that's not as good as a double blind study where you don't know what you're getting and I don't know what you're getting, and there can't be any bias there. So double blind is ideal, and all of the MOG and NMO studies so far have been double blind. I do want to emphasize the safety procedures. There are so many safety levels in place, from our site, from any individual site to central site. There's data safety monitoring committees that monitor the larger trial. I mean, there's so many levels of safety in place. That is really the last thing that I expect to fall through. And so one of the advantages of participating in a clinical trial is you know that there are many eyes on you. Everybody's paying attention to you. Everyone has their responsibility, that's true, but there are many safety processes in place to make sure that the drug is safe in you. So there are many phases of trials, and they don't have to go exactly like this, but this is generally true. First is a phase 1 study, usually like 10 or 20, usually healthy people, just to show that the drug doesn't kill you. These are very, very quick studies, usually 3 to 6 months, and then you're done. And then the FDA says, okay, drug didn't kill you, didn't kill any of the animals in the toxicity studies. You may proceed to phase 2. Phase 2 is where we try to figure out exactly how the drug works, what dose to use, and then how to monitor the outcomes. Because it may do one good thing, but not another good thing. You want to know what it's doing well, but then you design your phase 3 trial, and that's where you do the double-blinded, randomized, placebo-controlled, everything perfectly scientific to draw a conclusion. And then the drug gets approved, and it goes into phase 4, which is just observation. New drugs, new mechanisms. The FDA is like, you got to continue collecting data because we don't know what the long-term risks are. So those are phase 4 studies. Those are usually not blinded. Those are people on the drug, and we're just monitoring them for safety. Now, what control do you have in a clinical trial? Nobody has any control. You gotta follow the protocol. The protocol is key, because if you don't follow the protocol, we can't use the data scientifically. So I don't have any control outside of the study. Patients don't have any control outside of the study. Everyone's got to follow the protocol. But there's usually one exception to that. If something bad happens. Okay, so if something, let's say I give you the drug and you're in the trial and something really, you have a stroke and you end up in the hospital emergency room. The emergency room doc calls me. Everything is unblinded. You're out of the study. We give you all the data. Everything is unblinded because there's a major safety risk. Other than that, if you're like, I think I had a headache, or maybe I had a relapse, I really want to know what I was on, none of that can be revealed. You don't have any of that control at that point, and neither do I, because everything has to go through to the end, and then all of the data could be revealed. There is one other thing that you always can do. You can always stop participating. Once you're out of the study, again, you're free to do whatever you want. We'd never kick you out. We would never say, oh, you can't come to our clinic anymore. Never, never, never. You're always welcome. We love participation whenever you can do it. Had one MOG patient, he's older, 70s, flying in from another state every week into Boston. And at one point he was just like, I can't do it anymore. And I said, I appreciate it. Thank you for everything that you've given us. Don't feel bad about it. You've done a great thing for science. And he just, left the study. So that's totally, totally okay. That's always in your rights. So those are the nuts and bolts of clinical trials. And I think now we're going to get into details.
Thank you so much, Dr. Michael Levy, for that thorough presentation on clinical trials in MOGAD. We noticed that there were different phases of the clinical trials. Why is this important for us to know?
It's important because the level of flexibility, I think, is key here. In phase 2 trials, for example, there may be different doses. You may be randomized differently. There's just a lot more flexibility and many more phases of the fragment. The right wording is there's many more ways to participate, right? But once you're in a phase 3 trial, that's where everything is very, very strict and all the rules have to be followed. And I think, some people say, well, I really only want to participate in a phase 3 trial because it directly leads to an approval and I want to be part of that. And that's great. Earlier phase trials, they may fail because we don't know as much about the drug, but they're still so important to us. So we appreciate whatever phase you want to participate in, we're appreciative.
That's a great answer. Thanks so much. I want to ask Associate Professor Ramanathan this next question. As we're all aware, MOGAD, it's a relatively new disease. We have no really approved treatments, certainly no FDA approved ones in the United States. There's a couple of trials going on now, and there will be probably future trials. Could you tell all the patients out there and the caregivers, what are some of the benefits that these clinical trials for MOGAD would give patients and their caregivers?
Thank you, Julia, and thank you to the MOG Project for asking me to be part of this wonderful session. I think it's a really good question because in a newly recognized disease, the truth is we don't know the best type of treatment that there is, and it is a learning process. And a lot of the information that we have initially naturally comes from what we call retrospective cohort studies. And what this means is that you might have different centers in different countries that specialize in these kind of autoimmune neurological diseases. And they look at their own patients and they try and see, well, and the patients presenting like this type of treatment approach seemed to be more successful than another type of treatment approach. And this is quite an important thing to happen. It's valuable, it provides insights, but there are limitations to that in the sense that The treatments people use are dependent on clinician preference, clinician experience, their comfort with using certain treatment approaches. And so what you get is data that's a little bit murky and a little bit muddy, where there might be other variables that are influencing. Why did a person get selected for a particular type of treatment? Or why does that treatment appear to work? Do the very mild patients respond to a less significant treatment? Do the very severe patients unnecessarily get a lot of over-treatment? We're not able to tease these apart when we look at data that's collected this way. So that is an important phase in all conditions, and it provides insights. But then what you have is what Michael described, which is a really scientific approach of what is the specific question, what are the inclusion criteria, what are the group of patients we're looking at, and what's that predefined outcome. So the only thing that varies between patients who get the treatment or who get standard of care or placebo is the actual treatment, and everything else, like how severe their disease was, what gender they were, what type of presentation they had, all of that is taken out of the equation. And so I think what patients and carers get from trials is a very clear answer about whether or not a particular treatment is useful. The other benefit I think patients and carers get from a clinical trial is we had just heard from Michael about how patients are really closely monitored. These are trials that take their role very seriously. There've been a huge amount of logistics and administration to get through ethics. And I think the truth is a lot of these patients have very close contact with their trial team, with the research nurses and the research doctors. And while they might be blinded, they're very well monitored. If there's any concern about relapses, they get imaging. There's certain protocol that you have to undertake. So I think another benefit for patients is involvement and a sense of personalized management and care. And then I think it's also that concept of benefiting to knowledge about that condition.
Great. So we're really talking about benefits here, and I do want to follow up and ask, patients have their reasons that they may be concerned about entering one. In your experience, what do you think that you could outline some of the reasons that you hear from patients about why they might be concerned about joining one of these trials?
Absolutely. I mean, I think the reasons that patients are concerned are very, very easy to understand reasons. And if you just put yourself in the place of a patient, I guess the main concerns are, is this treatment that is the purpose of this trial really going to be better than the standard of care treatment? Am I being put on something that might be less efficacious than what's currently out there? The other big question is, would I be on placebo? Is there a period of time when I may not be getting any treatment? What does that leave me at risk of? And then I guess another question is this particular treatment under consideration actually safe for me? Are there going to be some side effects that have not come out somehow in the prior evaluation? And I think all of these concerns are extremely valid, very easy to understand and comprehend why they are concerns. And I suppose it is the onus is on the study designers, on the doctors who are choosing to participate on this study to try and ameliorate those concerns as much as possible. And I think the onus is on us to know that we have very significant preliminary data. We've done all the early phase studies, that we understand why we're doing something and that there's a really good reason. There has to be what I call clinical equipoise. There's got to be a reason we're doing this study. We shouldn't be doing a study because it's a no-brainer and we know this is going to work. We're doing this study because we're not sure of what its place is. And so there is a need for these patients may be exposed to unnecessary treatment if we don't do this study. Patients may not be getting the benefit of really efficacious treatment if we don't do this study. So you go in with a reason that we've backed up with preliminary data. We don't have the answer. We need this answer. But I think those are some of the concerns that patients have.
Thank you, Associate Professor Ramanathan, for highlighting the benefits and risks of a clinical trial. Since it is difficult to recruit patients who have MOGAD because of the low number of patients close to a site, Should patients always ask about whether or not patient costs are covered, for example, travel and so on? Are there any differences between industry-sponsored versus investigator-led clinical trials concerning reimbursement costs? This is an extremely hot topic, so we would like to take an American and Australian perspective on this. So Dr. Michael Levy, if you would like to start us off following Associate Professor Ramanathan.
Yeah, so MOG is rare, and we probably don't have enough clinical trial sites at every town to enroll all the patients that we would want to enroll. Everyone has to come in to a few sites, and that requires travel. And for these sponsored trials, these big phase 3 sponsored trials that are going to get the drug approved, there's a financial motive for the companies to pay for travel. And they do, and there's a stipend, I think it's something in the order of $500 or so to get here and get a room and get your infusion the next day and that kind of thing. So we have been able to get patients from all over. There was one at the beginning of one study. We were the only site open in the US. We had people coming, we had someone coming from Los Angeles to Boston. So even that distance was covered. Not the most comfortable trip. I'm not going to pay for first class. The hotel room is not going to be on the top floor. But I think to cover the costs, the companies know that they're not going to get all these MOG patients in Boston. So they have to be able to cover those costs, yes.
And I think, I mean, I agree with Michael from an Australian perspective. Australia is a huge country as well. We've got a lot of regional and remote patients. And so for industry-sponsored trials, often patients' travel accommodation is reimbursed. I suppose you asked a question also about investigator-led trials, and that's, I suppose, a different issue, and it depends on the independent trials, because the funding for an industry-sponsored versus an investigator-led trial is different. But I think wherever possible and wherever the budget allows, patients are encouraged to participate, and we have to provide them the support in order to do so.
Thank you, Dr. Michael Levy and Associate Professor Ramanathan, for your very important responses. Has there ever been a clinical trial that has been successful in recruiting patients that you have been involved in where travel and other costs were not paid? Dr. Michael Levy?
I can, we really try to make it available to everyone. I don't think patients are really looking for a source of income to participate in. Nobody's trying to make money from this. We're not trying to make money from this, but we really just want to make it available. So we've tried to cover parking. We've tried, whatever it takes to get the patients in and not be a financial burden. If they're really just too far, we may not be able to enroll. But I can't really think off the top of my head of a specific study, a specific trial where costs were not paid. We have to be able to make it available to patients.
Associate Professor Ramanathan, do you have any thoughts on that?
No, I mean, I agree with all of that.
Okay, I'd like to move on then and ask another question that kind of relates to what you were talking about earlier. And I'd like to ask both of you to get that US and international perspective. When a physician recommends, when should a physician recommend enrolling somebody, a MOGAD patient, in a clinical trial? And along with that, when should you not recommend somebody enter in a clinical trial, just based on your own experience with your patients? I think I'd like to start with Associate Professor Ramanathan.
Thanks, Julia. So that's an important question. I think when patients should be considered, I think the main thing is that there shouldn't be any doubt about the diagnosis. So you want to be enrolling patients who have the exact condition that you're looking to study. You don't want atypical patients. You don't want patients where we're uncertain about the diagnosis and we're considering that. And a lot of the study protocol and the inclusion criteria are geared towards making sure that we are looking at a specific question in a very carefully selected, curated group of patients. So I suppose that would be the first thing. We don't want people who are not meeting the pre-specified inclusion criteria. In terms of the patients that you wouldn't necessarily consider for the trial, I think once again, there are good exclusion criteria in many of these trials that are defined for a reason. Some trials don't start off in children because you want to see how it goes in adults. Pregnant women, it's often one of those population groups where we try not to enroll, depending on the trial, of course, but in these kind of conditions and these kind of treatment trials, we often don't want to have pregnant women because we don't know if impact on the fetus and so on. There are some patients who might have very brittle disease and who are very difficult to rescue, in whom certain clinicians might be very uncomfortable about the idea of a placebo. And I think the key in some of these trials is to make sure that we're not having patients who are very mild only being the ones who are in the trial. So there have to be pragmatic mechanisms where people can treat patients during a relapse in those first few weeks the way we normally would, before then taking away the extra treatment and keeping the trial drug versus placebo or trial drug versus standard of care. So I think trials also have to consider that we need to make sure clinicians are comfortable with how a patient is managed acutely. Other patients who won't be considered for trials are patients who might have side effects from being immunosuppressed. So a lot of the exclusion criteria, for example, eliminates patients who have active infection like latent hepatitis or tuberculosis or something, because immunosuppressing these patients can make those conditions worse. And then I suppose the other reason to not consider a patient for a trial is if they're not interested, or they're not likely to keep up with the follow-up routine or the follow-up schedule, because a lot of the investment in the trial is actually getting those predefined data points right up to the end.
Thank you, Michael. Do you have a perspective on this?
I would endorse all of that. I would also just add that usually I talk to patients, I tell them my experience with different medications, what I would normally use, that it's not scientifically validated, but these are the typical treatments that I would offer my patients. And then I also say, but there are also clinical trials, and these drugs, these mechanisms, would you like to hear more? And if the patient says, yeah, let me, I want to consider those, then I start to go into more and more details. And ultimately, I, of course, let the patient decide, and I'll say, among all these different options, I hope you're not feeling pressured to participate in the study just because I'm a scientist at heart. I want it to align with your priorities. What do you want to do? And the people at our site, for example, have all said, I want to be in the study. This is not something I feel pressured to do. This is not something that it's like my only option. There's never an only option. This is what I want to do. And so then I think that is the best way to enroll patients. It does obviously limit the number. I have about 200 something MOG patients, but only less than 10 or 12 so far have participated in any clinical trials. And a lot of it, I think, is the biggest drawback is the placebo risk. The chance that you're not, the 50% chance that you're not going to be on any treatment after having a relapsing course. I think that's probably the number one biggest deterrent.
So that's the kind of person that you wouldn't recommend. You would actually talk to that person and say, this just isn't for you.
I would never make that judgment that for anybody. I would always offer it I would always put it on the table. I'd say, this is always there for you. If you decide that you want to do this, I will help you.
Those are some good thoughts. Thank you.
Thank you, Dr. Michael Levy and Associate Professor Ramanathan, for those responses. I want to talk a little bit about the patient's responsibilities when they enter a clinical trial. From your perspective, what do you think these responsibilities should be, and how are patients made aware of these responsibilities? Associate Professor Ramanathan.
Thanks, Jenny. So I think that it is very much, as Michael says, how you put it on the table. And I think it's that question about, in a way, I sometimes think in our field, we could take lessons from other specialties where clinical trials are very much a part and parcel of management. And I think oncology is the one that comes to mind, where they really do move the field forward primarily based on these clinical trials. And their approach, speaking to oncology colleagues, is very different. And that becomes not to pressure patients and not to put patients in trial, but very much, as Michael said, put it on the table as this is what we think we could do. This is what we don't know about current options. Here are the trials that are happening to try and provide a more definitive answer. So I think the main thing is explaining the rationale to patients. They need to understand what the current treatment strategies are and why people think they are the way they are, what evidence there might be, what are the drawbacks to some of that evidence. I talked earlier about a lot of this coming from retrospective cohort studies where there are the confounders that might affect our interpretation of the data. So patients need to understand a little bit about the rationale about why we think this is a trial that is important. The second thing I think is that there's a very extensive process of informed consent that goes into trials, and patients, I always think it's never something you rush into. And if someone's interested in a trial, it's often a couple visits while this is percolating in their mind. They're getting information. They might go away, think about it, talk with the family members or friends or loved ones, come back with a few more questions. And I think giving patients that time to listen to your information, go away, think about it independently, talk about it to people that are important to them, and then come back with questions is a really important process of that, because everybody thinks of questions the moment they leave the doctor's office. And I think giving them the chance to reconsider and ask questions they might have not thought about at the time is really important. And then I think that the next responsibility is, so those are the responsibilities on the clinician when you're trying to consider someone for a trial. I think the responsibility on a patient is understanding what that trial means to them. Every trial has a patient burden, and by burden, I mean there'll be a number of blood tests, or there'll be a number of scans, or there might be a number of patient questionnaires that need to be filled in. And the reason these are very important are these are specific data points that have been selected as being important to understand whether or not this treatment works and the impact it has. And so patients should understand the schedule of visits, and a lot of these trials will have a really nice timeline about what's happening at each step of the way. And they have to feel confident that in their lives and with their additional commitments and responsibilities that they have the time to commit to this. Because I think when you enter a trial, it has to be with the purpose of seeing it through to completion, as far as possible. Of course, there are things along the way that might prevent that are out of your control. But I think everyone goes into the trial thinking that, well, this is something we're going to do. So I think it's just making sure that they have the ability in their life with their existing responsibilities to do that.
Okay, thank you so much. That's a great answer. So I'm thinking, about preventative treatments. I know somebody really has talked about that in the past, but, for these two clinical trials and the clinical trials that are coming up in the future, whatever those clinical trials might be, medicines like IVIG, prednisone, rituximab, and et cetera, how do you know when you're allowed to take that during a trial or not allowed to take that during a trial? Dr. Levy?
We'll tell you for sure. But for a lot, the two MOG studies that we're talking about, there's no allowance for preventive treatments other than the experimental drug. So you can't be on rituximab, prednisolone, IVIG. You can't stay on those drugs, because if, say, we give you a therapy and it works, how do we know if it's that or if it's the IVIG that you're using? So we have to make it clean. You have to be either on drug or placebo with nothing else to interfere with it. And it's just for a period of time, right? So say the drug fails or that you were in the placebo arm. That should emerge in the next few months. You might have your relapse, you get treated quickly, and then you're in the open label phase, and then you get the drug for sure. And then you don't need any more background therapy if you're getting the drug, presumably. So in that way, and then you don't even have to participate after that. Let's say you have your relapse in the study, and you say, I don't like this trial. I want to get out. I want to go back on IVIG. Great, you can do that too. But you just can't be on those drugs in the trial itself.
Thank you, Dr. Michael Levy. As you know, MOG antibody disease presents in, as far as we know, all different types of ethnic backgrounds and cultures. How vital is it to have diversity when enrolling patients in MOGAD or any other clinical trials?
So thanks, Jenny. I think this is a really important point, not just in MOGAD, as you mentioned, but just in clinical trials in general. And this is that it is very important to have diverse representation when you're trying to answer a specific question about treatment. I suppose from a scientific perspective, patients with different ethnic backgrounds and different genetic predispositions might even metabolize treatments or respond to treatment in different ways. And it's important to be aware of certain treatments that might have a different impact on one group of people compared to another. But more importantly, I think a lot of the reasons why a lot of effort is put in these large phase 3 international studies to try and be in as many countries as possible is so that the results of the trial represent an international population. And then you can use these results and say, look, this is an answer that we think is globally applicable, and this is an answer that we think can be used in patients of different backgrounds. So without having those patients enrolled in a trial that represent these diverse backgrounds, we can't really make that assumption.
Thank you for that great answer. I want to ask Dr. Levy, this is a pretty hot topic. I know we've talked before about monitoring patients for any health concerns. Could you tell us how important it is to prioritize that patient health during the clinical trial?
That's the whole point of the trial, is we want to prioritize the patient's health. And it covers everything. It's everything from a skin rash to a MOG relapse to just how you feel, fever, vital signs, blood tests, everything. Everyone is staring at your clinical record for the course of the trial, because for that period of time, that person's health is our top priority.
Thank you, Dr. Levy, for your important response. This next question is one of the most spoken about questions in the community and is pivotal for patients who are thinking about enrolling in a clinical trial. If at any point in the trial the patient feels like they're having a relapse, how essential is it for patients to directly contact the neurologist instead of going through the clinic? How does a clinic running the trial work with the patient's regular MOGAD medical provider? Associate Professor Ramanathan.
Thanks, Jenny. So this is a really important point because relapses are almost the thing that everyone's waiting for in these trials. So in the sense, many of the outcomes are time to first relapse. This is a key point of the trial. And I think the important thing to realize is that the treating clinician is blinded, the patient is blinded. And so all of these trials will have a trial team, and they will provide information to the patient about how to access the team as soon as possible if they're concerned about a relapse. So they will provide information that will enable the patient, if they think they're having a relapse, and they should provide a plan that if your relapse happens on a weekend, what do you do? Or if your relapse happens after hours, what should you do? Or if it happens during hours, who are the people you can contact? And the reason that's important is that usually we want to know about these very soon. As soon as they happen, we want to be aware of these potential relapses, because there are a number of things we'd like to do, like clinical assessments. We might want another scan. We might want further investigations. And then we often go through a process of relapse adjudication where we've already predefined what it requires in order to be counted as a relapse. It might be a step decrease in your visual acuity. For example, if you're having optic neuritis, it might be a change in your functional status. If you're having a cord involvement, it might be a particular change in your scan. So we need to know about this as soon as possible so we can do all of these investigations in a timely manner. Firstly, in order to make sure the patient can be treated with rescue therapy if we do indeed feel that that's a relapse that's happening. And then secondly, it determines what happens to the patient subsequently. So if that's deemed to be a relapse, as Michael said, the patient might then be eligible to go on. If it turns out they were on the placebo arm, they might then be able to go on to the open label arm of the active study drug.
Thank you, Associate Professor Ramanathan, for relaying how essential it is for the patients to directly contact their neurologist if they are experiencing a relapse. Speaking of relapses, how is a relapse determined in a MOGAD clinical trial, and how important is it to speed? How important is speed in adjudicating the relapse? Associate Professor Ramanathan.
So this speaks a little bit to what I'd mentioned earlier, where a lot of the discussion while you're writing the study protocol is actually how do we define a relapse? And this is important because people's threshold varies between different patients. So someone feeling a little bit off, or potentially another reason, someone might have a urinary tract infection and feel a little bit off their feet, or someone might feel unwell for another reason. And we don't want to misclassify that as a MOGAD relapse. And certainly some patients might have worsening symptoms that are very transient that then improve, that wouldn't necessarily be counted as a relapse. So one of the very important things to be very clear about in the study protocol is what are we going to accept as a relapse and what is required. Maybe it's an examination finding in a certain step decrease, or maybe it's a radiological finding, maybe it's a combination of both. So a relapse is determined by predefined measures that enable us to do this equally across the board to all patients who are enrolled in the trial. In terms of speed in identifying or managing a relapse, I'd say that it's very important. So patient coming to the trial team's attention is critical, and then the trial team acting on that to try and move things along quickly is important.
Okay, thank you. We have a community question I want to fit in. This person is asking if in any trials, are they contemplating creating a trial for a patient that has had only one single event? Does that even make sense in clinical trials? I think I'll ask Michael.
Well, we haven't rolled this out, but we are contemplating that idea. The idea that we are thinking is about half of people don't relapse. But if there was a way that we could make it so that nobody relapses, that would be great. So people would come in, they'd have their optic neuritis, get tested for MOG, and then maybe we could give them a drug of some kind that would ensure that they never relapse again. To be in that currently it's 50% that don't relapse. Could we increase the odds that you never relapse? So we do have some drugs in mind. We've approached one company in particular about a trial design for that. We have to power it correctly because we have to be more certain about those numbers. If half don't relapse anyway, we're going to have to enroll more people to be able to statistically prove that this new drug is working. But it is something on our radar, absolutely.
Okay, thank you. I wanted to also ask us another question, sort of switch gears. What if something actually catastrophic happens to the patient during the clinical trial, such as some severe side effect that, causes disability or permanent damage, such as loss of function? What repercussions usually does a patient have if something like that happens when they enter?
Are you asking me, Julie? It's never, I don't have personal experience of that with that ever happening. We've never had a case where somebody comes in and due to the study drug, they've had some permanent damage, neurological or otherwise. I haven't had that personal experience yet, so I don't know for sure. What I can tell you is the company and the insurance and the hospital and everyone involved in your care is going to jump on top of treating you right away. And I think that's one of the advantages of participating in a study is you get so much attention from everybody all the time that we really think if something bad does happen, we're going to really try to capture it as early as possible. So that I think is one big advantage. The disadvantage is you're trying a new drug and maybe it's never been in human before. Maybe it has, but in a different disease. And so there is some risk involved in that. Usually, I'd say before we even get to a phase 3 study, we've done the phase 2. We have some feeling of how this drug would work. And so we're not just going to throw bleach into you and hope that it doesn't hurt you. We really are thoughtful about what we're trying. So I don't have a great answer. Maybe, Darshi, have you ever had an experience with a really, really bad outcome from a drug like in a
No,
trial?
not personally. I mean, we've had patients who've had side effects that we were thinking would happen with that particular condition. And then, as you say, we're always monitoring for it. We're always on the lookout for it. So it was picked up quite early. It was then managed appropriately. The patient did fine. Patient wanted to stay on the study drug despite that, because they were quite happy with the fact that their clinical disease related to the condition was so stable, which they hadn't had before. Well, they didn't know they were on the study drug, but it turns out they were, but they could see a period of disease suppression outside of that complication. So with, touch wood, not had a catastrophic event, but I think I would echo what you say. You're so carefully monitored that the hope is that these things are picked up early. The other thing is that bad things can happen unrelated to the study drug. Patients may have been having a stroke or a heart attack anyway, but we assume everything could be connected to the trial they're on until we can definitively exclude it. So we'll never just take things off the cuff and say, nah, probably not related. So I suppose that's the other side of it.
Yep.
Thank you, Dr. Michael Levy and Associate Professor Ramanathan. Who is involved in the analysis of the clinical trials data? For example, is it just one doctor in one country, or are there multiple doctors in multiple countries, or is it just the sponsor and the pharma company who evaluates the data? Should an outside authority review the data in every case? Associate Professor Ramanathan.
That's a really good question, and it speaks to the fact that so much of conducting a trial scientifically depends on all of the pre-work. So you can't just get the data and ad hoc look at it how you will. Before a trial is even started, you have to identify what is your exact question you're answering. How are you going to power the study, by which I mean how many patients do you need to try and find out this estimated effect size change between the study drug and either placebo or standard of care. You already have to have a statistical analysis plan in place. So you already have to have had your independent biostats expertise into the grant to say, if we get this many patients in this trial recruited, here is how we're going to look at the data. These are the factors that we're going to use to make sure that the data is equally divided between these two study patient groups or study arms. So a lot of this is pre-decided. And the really important thing in trial is you can't change it. You can't change the way you analyze the data depending on what came out in the trial. If you did not reach your primary outcome measure based on your predetermined statistical analysis plan, too bad. That is the answer for your trial. So a lot of the work has to go into it before the trial is actually even started. And then you are bound to continue down the path that you've decided you would go into. So a lot of forethought and planning is there. You also have, your biostats analysis is quite an independent group, and you've already determined the individuals who are part of this. So it's certainly not anyone who might have vested interest in this particular thing, or any clinician who has a very strong opinion about this. These are biostatistics experts who are given a particular plan or have come up with a particular plan in order to analyze the data. And I think it's really important for people to know that the data is what the data is, and there's no subgroup analyses or ad hoc analyses that are allowed in order to try and swing results one way or another.
Okay, thank you so much. I want to ask this one of our last questions to Michael Levy. Given the fact that there's so many patients, there's only so many patients that can be involved in the clinical trial because MOGAD is just such a relatively new and rare disease. How can we be sure that the results are going to hold the same accuracy? Some of the other drugs that are more common in other diseases such as MS and in something like that, there's a lot more participants.
Yeah, so after the trial is done and we have our scientific data, it's going to be a small sample size. We're going to be comparing 50 versus 50, something like that. So that is a valid concern. And so what we're going to do after that is the phase 4 study, where we track people who are on the drug for many years, hopefully years, and not just keep track of the safety, which we want to know, long-term safety, but we also want to use this kind of real-world data, because they're not going to be in a clinical trial where every little detail is going to be recorded. They're going to be in an observational study where they might miss a dose, they might forget a dose, they might take 2 doses. Real world type studies are not as perfect. And we want to make sure that even in that real world scenario, that the drug efficacy and safety matches what we found in the phase 3 studies. Then after those phase 4 studies are published, it gives people confidence that what they found in phase 3 is not just scientifically true, but is practically true. And so those phase 4 studies are really important as well.
Thank you, Dr. Michael Levy. So every year we continue to learn more about MOG antibody disease and research that evolves. What exciting research is being done for MOGAD that you are working on? We are excited to hear from both of you. So Associate Professor Ramanathan, if you would like to start us off, followed by Dr. Michael Levy.
Thanks, Jenny. So I suppose our group is quite interested in 2 arms of MOGAD. One is from a pathogenic perspective, why this condition occurs in particular patients, what are serological biomarkers of disease activity or prognosis, what are clues that we can use to help identify at disease onset what a patient might do, and whether or not a patient is more likely to be relapsing versus not, whether a person's more likely to have severe disease versus not. The other type of research we're very interested in is clinical research, and how best to improve diagnosis and separate these patients from those who have ACOPOIN4 antibody positive disease or multiple sclerosis, how to improve our understanding and interpretation of results that are done by varied assays, so patients aren't missing a diagnosis of MOGAD or incorrectly being given a diagnosis of MOGAD. And then particularly with treatment, we're very interested in data about what we do at treatment onset, how we manage these patients at treatment onset, how does that impact or potentially modify their disease course? And some of the work that we're doing currently under review suggests that perhaps there's a way of treating patients when you suspect them to have the disease that might reduce relapse rates in those first 12 to 24 months. So trying really to optimize treatment at onset. And then, of course, in a person who is a chronically relapsing patient, what are the best approaches there? Thanks, Jenny.
My turn. We're doing studies in 2 areas that I think are really exciting. One is trying to understand what triggers the disease in the first place. So MOG is a protein in your brain. Why on earth would your immune system not accept it? Why does it want to attack MOG? Are there other proteins in your environment, maybe in viruses or in your diet or something else, that triggers the immune system to attack MOG? So we're really interested in that question. And then the second relates to the idea that you could re-educate the immune system, this idea called tolerance, whereby right now your immune system doesn't like MOG, but can we get your immune system to re-accept it? Can we just switch off the immune response to MOG in the same way that your immune system turned on to MOG? Can we just switch it off? So we're looking into ways of doing that. There are companies interested in that. We're working together with them. They have different platforms, they call them, different mechanisms, different ways to get the immune system to accept something. So we're hopeful that since about half of people after a MOG attack never have a second attack, they just retolerize. Their immune system says, you know what, I apologize. I didn't mean to attack MOG. I realize you're friend, so we're going to reaccept you. Can we generate therapies that basically reproduce that same process? So that's what we're really excited about.
Yeah, both your research sounds fantastic. We're really excited to see what comes up in the future. It's pretty lucky days for the MOG patients, I gotta say. So I just want to say a massive thank you to our amazing experts, Associate Professor Darshi Ramanathan and Dr. Michael Levy. You've taken the time to help us understand all this about clinical trials in MOGAD and around the world, and the significance, and what we're just one step closer to FDA approval here in the United States for something. We're really excited about that. So I really appreciate your time.
Thank you for inviting us.
Any unanswered questions will be collected, answered by the doctors, and made available in a follow-up MOG blog for this event. This podcast will be available on our YouTube channel also. Thank you so much to the both of you.
Yeah, thanks for this great discussion. We wish you luck in your future research, and have a great rest of the day.